Hello, good morning, good afternoon, good evening. I'm Paul McLaughlin. I'll be emceeing this session again. I am at Ericsson, and I'm really happy to introduce James Ashenhurst and Shannon Hamilton, both from 23andMe. And they will be speaking about or giving a presentation titled Discover, Predict, Prevent, Developing and Delivering Personalized Polygenic Scores at Scale. Just a reminder, the presentation will last until 12:05 when we take a lunch break. So I encourage the speakers to leave about 5 minutes at the end for questions. And if you have any questions, please put them in the chat and I will read them out loud at the end. Thanks so much. Take it away.
Thanks, everybody. Sorry for that little glitch, but I'm a product scientist at 23andMe, and I'm going to tell you about a collaborative effort between scientists and engineers to build machine learning infrastructure to support rapid development and enhancement of the 23andMe product. So a little bit more about 23andMe. If you hadn't heard of us before, our mission is to help people access, understand, and benefit from the human genome. And we do this through a direct-to-consumer genotyping kit, where you order the kit online, it's delivered through the mail, you spit in a tube, provide a saliva sample, we process the DNA, interpret the results, and return the results to you through a web portal. The kinds of reports we offer span from more serious health predisposition reports, which include FDA-authorized genetic health risk reports on serious topics like Alzheimer's disease and Parkinson's disease. Also, FDA-authorized carrier status reports, which are particularly relevant for family planning, informational and educational trait reports, and we can tell things like, based on your genetics, the likelihood that you have unibrow. And lighter health topics in the wellness category related to things like alcohol flush reaction or lactose intolerance. And then, of course, our flagship ancestry composition report, where we can estimate the ancestral origins of your DNA. And here is an example of a health predisposition report. This particular one is about type 2 diabetes. So what we're telling you here is, based on your genetic profile, the chances that you might develop type 2 diabetes between your current age and some future date, like say age 80. And importantly, this report is powered by a polygenic risk score, which I'm going to explain a little bit where that comes from. And it's also powered by 23andMe research. So the algorithms that power these results are actually developed internally and based on our research platform. As if you didn't know, 23andMe actually has one of the largest genomic research platforms in the world, where our customers can actively opt in to participate, answer surveys, and we can combine those survey responses with their genotypes and make discoveries of association to try and understand some of the origins of the heritable differences in traits, or also disease likelihoods. And very importantly, 23andMe research is an opt-in system where customers actively consent to participate. And all of our research protocols are independently governed by an IRB with oversight and approval over our protocol and consent processes. And customers can end participation at any time. And as part of human subjects research, all data are aggregated and de-identified. But what we really see this platform doing is being part of this virtuous cycle, where we create educated, engaged consumers who are learning more about what their DNA can tell them about lighter topics to more serious topics. People want to participate in research, provide survey answers. We can make discoveries, which we then can turn around and put back into the product and provide more insight and more value to our customers in this cycle of discovery. And so far, we have over 4 billion data points in our research platform, and this comprises some very, very large cohorts of both common and rare diseases. So for example, we have well over 1 million people who self-report a history of diagnosis of major depression, or we also have more rare things like scleroderma or sarcoidosis. So we're really quite powered to make discoveries of what genes may underlie vulnerability to these conditions, and we can turn that around and provide insight back to our customers. But in order to really understand the technical challenge that we need to solve, it's important to understand the science behind some of the genetic causes of heritable disease. So what we're looking at here is a commonly observed relationship that we see between how common a variant is, a genetic variant is in the population, and how big of an effect it has on the chances of developing some disease or outcome. So on the x-axis, what we're seeing is how frequently a variant is observed in the population. So that might be a change from a G to a T at one specific location on a chromosome. Some of these variants are rare, and some are a lot more common. And on the y-axis, we're looking at the effect size or odds ratio, or basically how big of an impact does having that variant have on the likelihood of the outcome. In the top left corner is a cluster of disease variants called Mendelian disease variants. You might recognize the name Mendel, Gregor Mendel, in that name. That, of course, is referring to the German monk who was breeding peas, and probably what you remember from genetics and high school biology, where single genes seem to have a deterministic effect on the outcome, and variation in those genes cause changes in these outcomes. There can be dominant or recessive patterns of inheritance, but having one or two copies of the variant means you definitely will have the outcome. An example would be something like variation in the CFTR gene in cystic fibrosis. Disease-causing variants in these genes, if you have one or two copies, definitely will result in cystic fibrosis disease. In the kind of more middle spectrum, there are variants that are more common in the population. The cystic fibrosis variants are actually very, very rare, but there are variants that are more common and have kind of more of an intermediate effect on the likelihood of the outcome. An example there would be variants in the F5 gene, as they relate to hereditary thrombophilia, which is a blood clotting disorder. So these variants are more common. Some of them are around 1% of the population has them, and they increase the chances that you might develop harmful blood clots in your lifetime, but it's not a guarantee that you'll develop those harmful blood clots. In the lower right category, we have common diseases, which are explained not by single variants of large effect, but many, many genes of tiny effect. So for common conditions like type 2 diabetes or heart disease, these are explained by hundreds to perhaps thousands of genes, each of which has a tiny effect, but in aggregate can give us a picture of your broader risk. So how can we assess the contribution and aggregate all of those individual effects into one algorithm? We do this in what's called a polygenic risk score. So as the name implies, it's multiple genes. And the process of developing these at 23andMe starts with data collection, then doing what's called a GWAS, a genome-wide association study, followed by modeling and developing candidate polygenic risk scores, and then implementing those into the product where we can return personalized risk estimates to our customers. So at the start with the data collection, we have research participants who've opted in and they provide DNA and they have responded to surveys on various topics. We put these survey data through scientific review and quality control them to make sure that it's capturing what we intended to capture, that it's capturing the disease concept that we want. And our data collection team spends a lot of time crafting surveys and language to make sure that it's really a legitimate representation of self-reported disease. We then take those data and we do what's called a genome-wide association study, or GWAS. And this identifies genetic variants that distinguish people who have the condition, say type 2 diabetes, from controls, people who do not. And we do this in a training set. A typical training set that we have at this point can be around 1.8 million people. So we have about 1.8 million people who have answered this question, and we have their genome-wide genotyping data. The result of the GWAS, I'll explain a little bit more detail of how it's conducted, is measuring the statistical evidence for the association between every single location that we've tested on the genome and the outcome. But this doesn't capture the aggregate effects altogether. So in effect, the resulting summary statistics from the GWAS is what we use for feature selection to build our polygenic models. The GWAS itself is not the feature selection process. So what goes into a GWAS? And these are the bread and butter of what we do very routinely at 23andMe. So key ingredients are having genome-wide genotyping data for all the participants who are involved. So the genotyping chip that we directly test participants on has around 500,000, 650,000 genotypes spanning across the genome. So we have that many directly assayed data points per person. And we have the option of making better guesses and imputing out to, say, 45 million genotypes across the genome based on imputation relative to reference panels. So that means we have between 500,000 and 45 million genotypes per person across that training set of 1.8 million individuals. We then do a series of parallel linear models to regress the outcome as a function of each genotype, potentially controlling for other demographic factors like age or sex, if those are important. And then we also routinely include genome-wide principal components, which can account for population substructure. So in effect, what this means is running massively parallel computing with up to 45 million plus regressions on over 1 million people. So this is one step that requires a lot of computing power, and then all of those aggregate statistics are put together and are ripe for figuring out what genes might be associated with the outcome. So what you're looking at here on the left is one common way to display the results of a GWAS. It's called a Manhattan plot because it can kind of resemble a city skyline. So on the x-axis, you're looking at chromosomal position from the first chromosome up to the X chromosome, which is the 23rd. And then on the y-axis, you're looking at the negative log 10 of the p-value of the beta of the association between the variation at each one of these locations across the genome and the outcome. In this particular GWAS, we're looking at the results for caffeine jitters. So this is, do you tend to be a little jittery when you drink caffeine? What we're seeing here is more of a monogenic pattern. It's looking like caffeine is majorly driving the heritable individual differences in caffeine jitters. And it turns out that this gene actually encodes an enzyme that is part of caffeine metabolism. So there's a story there, a biological story that makes a lot of sense. Other GWAS result in Manhattan plots like this. So they really much more resemble a city skyline. So here for hypothyroidism, we're seeing hundreds to maybe thousands of genes that have statistically significant association with the likelihood of the outcome. So this sort of a GWAS result set is very ripe for building a polygenic risk score so that we can identify all these variants, aggregate all their effects, and estimate individual likelihoods given an individual's genetic profile. So we do this by aggregating, as I said, the effects of the individual variants into this PRS, polygenic risk score. And this really is jointly modeling all of the different variants that we think might be involved in, say, logistic regression, if it's a binary outcome. We might also have age or sex terms, or maybe an age squared term if there seemed to be a quadratic relationship with age. And all of those individual effects are summed together into the single score. Typically, in the models that we're developing these days, they include around 100 genetic variants to up to 25,000 genetic variants that appear to be associated with the outcome. But an important question is, how do we choose which of these variants from the GWAS to be included in a model? So it's a feature selection problem. And really how we're thinking about this is balancing the signal to noise ratio. So we want to maximally get all the predictive power available from that we're seeing in that Manhattan plot. But there's also a correlational structure across the genome called linkage disequilibrium, because variants that are physically closer to each other on the genome tend to be inherited together. So that means that variants that are next to each other tend to be highly correlated, and thus their associations are both, variants that are next to each other are likely to both be associated with the outcome. So we'd like to balance the signal to noise and reduce our features so that we're really maximizing the predictive power. So there are a couple parameters that we can permute to nominate SNPs, single nucleotide polymorphisms, variants, to be included as genetic features in the model. And these can be, what's the maximum p-value that we might consider to be included in the model? Or what's the minimum distance that we might have between variants for both of them to potentially be included in the model? So we vary a bunch of those hyperparameters, and we might have a dozen to maybe 100 different feature SNP sets that we then would test. So we would use the training set that was in the GWAS, and we test these dozen different models, dozen different SNP sets, and we assess their performance in a validation set, which is typically around 200,000 people at this point, to see which of these feature sets performs the best in terms of things like area under the receiver operator curve is a key statistic we look at. And that way we can pick the specific feature set that works the best and promote that for use within the product. In this final step, to do the final assessment of how well the model works, we assess it in a test set, usually of about equivalent size for the validation set, that was not part of any of the upstream analyses. So they were not part of the GWAS, they were not part of the model training, they were not part of the model tuning. So in that way, it's less biased and less prone to perhaps overfitting. And the statistics that are derived from our assessment in the test set are what we report in any white papers about the performances of these models and in any customer report information that we provide in the product. And then finally, these polygenic risk scores are interpreted into the consumer-facing report. So then we can do some epidemiological statistics to say, like, people at a given percentile in the distribution of polygenic scores have an X% likelihood of developing type 2 diabetes by age X. And that's the result that we return to the customer. But another important point I'd like to make is that 23andMe believes it's important that everyone benefit from genomic research, and we do strive to make PRS, polygenic scores, that work for everyone. Because unfortunately, genomic research has a diversity problem. As of 2016, when this study came out, a majority of the data that go into genomic research studies are from people of European ancestry. In 2016, it was estimated that about 80% of those studies involved people of European descent, the next largest group being those of Asian ancestry. This is obviously not representative of the genomic diversity of the world, and we do want to make sure that people have the opportunity to participate in research and to ensure that they reap the benefits and that the models all work well for them too. This is particularly a problem in the context of polygenic scores because models that are trained in one population sometimes do not generalize well to others. So say if we go through that entire flow of identifying a cohort, doing the GWAS, training multiple candidate models in parallel and promoting a model, if only European individuals were involved in that entire process, and then we test how the model works out in, say, an African American population or an East Asian population, it might not perform as well. And why is that the case? Well, so that correlational structure that I was talking about across the genome means that the models that we are producing likely do not contain only causal variants. So that substitution of an A to a G at a particular locus might not actually change the function of the protein that results from that gene. It might not actually change the expression of something. What it's doing is tagging a nearby marker that is actually the causal variant. So given this correlational structure, they don't necessarily contain causal variants. And these correlational structures tend to vary between populations. So this very rich diversity that we see across populations is what we can use to power things like ancestry composition and make estimates of where in the world different segments of your DNA originated. But that kind of variation confounds and can obscure our ability to translate polygenic risk estimation from one group to another. So how we're addressing this now at 23andMe is to do automated population optimization at every step possible. So what this means is considering if there is sufficient sample size, we could perform the GWAS in multiple populations. Say if there are about 5,000 cases, that tends to be enough. And then we can combine the results of those multiple GWAS into a meta-analysis. And the summary statistics derived from a meta-analysis tend to nominate more robust variants for inclusion. They tend to be variants that work better in various populations that we might test them in. Additionally, we can use transpopulation megacohorts in training, and we can optimize our feature sets in population-specific validation sets to make sure that any candidate model that we might try will be tested in every different population that might receive it, and we can optimize which specific feature set goes to which particular population. Then we calibrate the model interpretation to increase the accuracy. So now that I've told you about the scientific challenge we were facing, I'm going to hand it off to my colleague, Shannon Hamilton, who will tell you about the engineering challenges we faced to develop these at scale.
Thanks, James. Can you hear me okay? Awesome. All right, hi everyone, my name is Shannon Hamilton. I'm a product manager at 23andMe and excited to talk to you about PRSmachine, a piece of infrastructure we built to be able to deliver these cool models that James is building to our customers. Next slide. Awesome. So with PRS models as a real cornerstone of our scientists and engineers needed a better, faster, and more scalable way to easily train, validate, and deploy our models to customers. So we built a new machine learning service that we've lovingly named PRSmachine. And there were three main goals for this work. So first, thinking about it from the company product perspective, 23andMe just launched a new subscription plus product. And so we wanted to be able to launch new PRS-powered health reports more frequently to our new subscription customers. Now looking from more of an infrastructure point of view, sorry, if you can go back, that's okay. We wanted to be able to build, train, and ship models in the same environment. So the main goal of this is to ensure that no complex cross-team handoffs between scientists and engineers, and it would remove the need for time-intensive model validation that's required from training one model in research and then deploying it in production. And so we wanted to be sure that the exact model object generated in training is used for actual prediction of our customer results. And so being able to do this would effectively shorten the development window of our PRS models down from months to hours, which would be a huge win. And we also wanted to be able to predict our customer results on the fly. So this was important to us so that we could avoid unnecessary storage costs of large amounts of data, removing the need to pre-compute any of our results, and ensure GDPR and CCA compliance as well. Next slide, please. Thanks, James. So now we've told you a little bit about the why, I want to jump into the how and explaining three ways that we believe PRSmachine improves our development workflow. And the first I want to focus on is this first point that we believe our PRSmachine really centralizes and standardizes model tracking for our scientists and engineers. Next slide. And the way we do this is via an open source tool, MLflow from Databricks. And so we all know, I'm sure there's many data scientists in the room, that experimentation is key to this data science workflow. We want to try different feature sets, model parameters, packages, solvers, and with the end goal of achieving the best model result. And so MLflow really allows our scientists to track and store their models, the artifacts, the metrics in a transparent and organized way. So scientists like James organize their modeling work into experiments and runs, and then the model objects that are generated are stored and referenceable for promotion and serving results to customers later as well. So the great win of this is that it reduces errors, removes the messy headaches of keeping track of Jupyter notebooks, and really promotes transparency across the organization. Secondly, we believe PRSmachine really improves our model development workflow via enabling co-development by our scientists and engineers. Next slide. And the way we do this is via Metaflow. So at 23andMe, we believe it's really important that our product development comes from scientists and engineers building together, often in a single repository, committing production-based code and in a way that really, also enables the scientific experimentation that's inherent to data science. And Metaflow really enables that. So it's a Python library built by Netflix, also open source, that really allows scientists and engineers to string these modeling steps together into workflows. And because it's Python-based, the programming model is super intuitive to our data scientists. So no need to learn new workflow languages or operators, and removes any friction to the adoption of the framework too. So it's something we've been excited to integrate into our workflow.
Shannon, I'm so sorry to interrupt. We are actually at time for the panel. So I just wanted to note that we do have a lunch break coming up.
Okay.
So people people are welcome to stay. I know that you probably have a bit more to present, so I would encourage you to presentation, and I will be happy to stay with you and read out questions, but we may lose some people because we do have a lunch break coming up until 12:45, and then we will be back with another presentation continuing on in this theme. But so please continue. I just wanted to let people know that if they do want to get some lunch, please feel free to step away as well, and we'll be back at.
Perfect. Thanks, Paul. Great. So this is just, now we're talking about Metaflow a little bit, this is just an example of how we break up our pipeline, as James described, all these steps into individual step functions within it. So just a quick example. Next slide. And then lastly, and most importantly, PRSmachine really improves our model development because it enables the delivery of more robust and meaningful insights to our customers. Next slide. And there's two important pieces of infrastructure that we'll talk about briefly that allows us to do this. So something that we've called an interpreter. And I also want to speak about our prediction endpoint next. So we'll start with the interpreter first. Every model trained is kind of through our pipeline using PRSmachine is paired with an interpreter. So the interpreter contains result thresholds and then does some complex statistics to really produce the customer-facing result. Next slide. So to make this all tangible, here's an example of what a customer might see on our website or in our app. And so, for example, we just released a new health report to communicate an individual's risk for coronary heart disease. The PRS model that powers this looks first at an individual's genetic data, then other data like age, sex, and ethnicity, and then predicts and outputs a risk score for that particular phenotype. This score is a raw number in a super arbitrary scale, and the interpreter will take that number, interpret the risk score, and transform it into something meaningful for our customers. So you'll see, like on a page, the actual risk of acquiring a disease like coronary heart disease by age 70 communicated to our customers ultimately. Next slide. Thanks. And then in addition to the interpreter, the endpoint design is another way that we believe allows us to serve more robust and meaningful insights to our customers. So the way that PRSmachine interacts with our production repository is via this prediction endpoint. And so again, like I said earlier, in order to reduce storage costs, optimize for GDPR and CCPA regulations, we wanted to be sure that PRSmachine is truly performant enough to compute a customer's result on the fly. So this means anytime a customer visits a given report in the website or app, their result is computed in real time. And this, though, the ability to do that is dramatically affected by how big the model is, or how many different locations on the genome we use for prediction. And so this slide shows a latency plot that shows how long it takes to get a result for a customer given the size of the model. The pink arrow points to where our largest model is at currently. And so one benefit of building PRSmachine is that we're now able to serve results for much bigger models now. Some of our early reports in the app were used only 300 different locations in the genome for prediction. And now our most recent points are using up to 25,000 different locations. So it's a huge win. And we're really excited about our increased ability to generate more accurate models for our customers and hopefully provide even more meaningful results. That's it. And just wanted to, yeah, perfect, James.
Yeah, so I would definitely want to thank research participants, employees of 23andMe for making this possible, and some of our leads, Derek Kondo and Shiva Amiri, and the different engineering and science teams, without whom this would not be possible.
Well, thank you so much for the presentation. Are there any questions in the chat? Feel free to post some questions for our speakers. I thought it was a very interesting presentation. Unfortunately, you all may be competing a bit with lunch and coffee.
No problem.
Thank you so much for joining us, and have a great rest of your afternoon. We'll be back after lunch at 12:45.
Awesome.
Perfect. Thanks, Paul.
Thanks, everyone.